Hi everyone, my name is Robert. I'm privileged to be CEO at Vor Bio. We're a cell therapy company who is public, and you'll be able to read the disclaimer slide at your leisure on our website. So who is Vor? Well, we are a kind of unique cell therapy company. We'll describe a little bit about our approach in a little while. We are working on two different modalities, both HSCs as well as CAR-Ts. We'll show you a little bit about the clinical data that we've received to date, which is super exciting and really holds the promise that we could potentially transform acute myeloid leukemia, which is really a devastating disease for which we need many solutions. We're privileged that we actually even built out a modest GMP facility in our own space. It happens to be in our own office space in Cambridge, Massachusetts. It's 4 clean rooms that are equipped to make all different kinds of cell therapy products. And we're relatively well capitalized with 187 million as of June. So let me just get into a little bit about the history of Vor. Vor, in case you're wondering, is the name of the Norse goddess of wisdom. Before you ask, that was not my choice. The company did something very dodgy in 2019 and hired me, but we've been pretty successful at raising money. We did an A round of 42, 110 million B round, IPO'd in 2021, and then raised our first successful public follow-on end of last year. We'll talk a little bit about this in more context in terms of, how to survive this downturn a little later on in this presentation. But I think one key advantage is that we've been able to make a lot of progress. So we've been moving forward rapidly with what we now call Trem-cel, which is a gene-modified stem cell transplant for patients with cancer, and generated some clinical data on that. And then we've just recently gotten the second program into the clinic, allowed our IND called VCAR33 Allo. And that's a program we're super excited about, which provides not just an interesting opportunity for patients with AML, but an ability to synergize potentially with what we're doing with Trem-cel. So let me just talk about our first approach, which is what we call shielded stem cell transplants. Now, in terms of what generally happens in AML, lots of people pursued AML for a long time. AML is a cancer which actually reliably expresses certain cell surface proteins that include CD33 and other targets. Unfortunately, as you know, cancer is derived from self, and these cells molecularly look almost identical to stem cells. And so the issue is that you can target these cancer cells very effectively. Unfortunately, that means you're also targeting healthy cells, and you happen to need a bone marrow to survive. So the revolution that our founder, Siddhartha Mukherjee, came up with is, what if you stopped paying attention to the target? What if you paid attention to the healthy cells? Because these cells in a transplant are taken out of a healthy donor, and they're outside of the body and then put into the transplant recipient. What if you could manipulate these cells in between and somehow shield these cells from targeted therapy? And that's exactly what we do at Vor. And so I'm showing you some data, some clinical data from a first-in-human clinical trial where we edited the stem cells for AML patients. And on the left, what you see is that in the first 5 patients we treated, we were able to successfully gene edit 80 to 90% of their entire CD34 population and remove out a gene called CD33. So firstly, I think we can very successfully, at scale, manufacture a healthy cell product that is missing a particular gene called CD33. Now, what does CD33 do? Goodness knows, I don't even know. But what we do know is that we felt it was possible that this was biologically dispensable. On what basis? Well, animal models showed that, but there's also about 1 in 10,000 people in the general population walking amongst us who have never, ever expressed CD33 in their entire lives. That's kind of interesting. And so our thought is maybe you could edit this gene out, and then we should see what happens to these cells. Do they behave like regular stem cells? And what you're seeing on the right is a chart which is the primary outcome of this study. We expect these cells to engraft like any regular stem cell. And it just so happened to play out that way. So fortunately, these patients had a post-transplant journey that is to be expected. These cells magically migrate into bone marrow. They start proliferating and differentiating, including, of course, neutrophils, which are really vital cells that protect us from bacterial infections. In fact, this is the #1 reason why patients can be discharged home after a transplant. And you can see in all 5 patients, these cells engrafted normally, in fact, even ahead of our benchmark of 11 days. And so we now have some clinical data that bears out this question that it does seem that this gene is potentially biologically dispensable. What we then did for these patients is we made a drug available called Mylotarg. This is a drug marketed by Pfizer. It's an antibody drug conjugate that targets CD33. Now, so this drug Mylotarg is not actually that often used, primarily because it's so incredibly toxic. Anything targeting CD33 will decimate your heme system, make you pancytopenic. But in this case, the first patient who received Mylotarg got 3 subsequent doses of Mylotarg, and you can see from the cell counts that it didn't even touch their blood system. So at least in this first patient, we bore out that we could protect their heme system from what would normally be a toxic dose of Mylotarg, and their cell counts were entirely preserved. How do we know it was potentially toxic? We measured pharmacokinetics, which is what you see on the right side. And despite the low dose of Mylotarg we gave them, it behaved on a PK basis like a higher dose. And that's because they simply had no antigen sink around. We deleted it from their bone marrow. And so this dose behaved almost like 8 to 10 times as high on an AUC basis than the low dose that we gave them. So it certainly should have made them cytopenic in that regard. So this was an N of 1, of course. We need to bear this out in additional patients, but at least in this patient, we did seem that we could protect their blood system. So let me talk to you about our second approach, which is a CAR T. And it's like, I can see you guys going to sleep right now, but please bear with me. The CAR T that we're doing, we know all about the difficulties with autologous therapies. They have revolutionized the world, particularly in B-cell cancers and plasma cell cancers. Unfortunately, in AML, autologous cells, these patients are super beat up. It's different when you have a peripheral disease versus a bone marrow disease, and you're getting deep chemotherapy that affects the phenotype of their cells. Secondly, we know some of the difficulties we've seen with allogeneic cells, where there's difficulty in getting these cells to persist long-term or proliferate in vivo. We're doing neither of these things. What we're doing instead is we are taking patients who relapse after their first transplant, and we're going back to the transplant donor and asking for T cells. Now, there's a few different advantages here. Firstly, these donors have never seen chemotherapy. They're by definition healthy. Secondly, there's a ton of these cells available because, again, they are healthy people. But thirdly, which is what's super fascinating, is that their immune system is essentially identical to the transplant recipient. I heard a joke from a transplant donor, from a transplant recipient who said, you know what, I better not go and murder someone and leave blood evidence because they'll arrest my donor. And that's actually true. They have the same blood system. And so what if you were to take these cells, which are identical in HLA, and use those as a starting material for CAR T. So that's exactly what we're doing. This is a protocol that is currently enrolling patients. We received IND clearance mid this year. And what we're able to do is take patients who have relapsed and treat them with a CAR T derived from their healthy transplant donor. Of note, in this protocol, we asked FDA's explicit permission if we could not just dose regular transplant patients, but also dose TREM cell patients from the first clinical trial that I showed you. And FDA agreed. So this means that we could potentially, in this study, we could potentially see the synergy between a shielded bone marrow transplant and a living CAR-T drug that pursues the same target. We think this might be a pathway towards a potential cure, and this is something we'd love to interrogate as patients enroll. Lastly, let me just talk a bit about manufacturing and get into generally what our environment looks like. So we've invested a lot in terms of process development and analytical development at Vor. We have the vision that we can actually generate not just an HSC product, but also a CAR-T product from the very same apheresis product from a healthy donor. It just so happens that when you take cells from a healthy donor, aside from the CD34 positive stem cells, you get a lot of other cells that include T cells that could be an ideal starting material for a CAR-T. We've been privileged to actually generate a small GMP facility inside Vor, and we have operators and on-site quality people who can produce and release material. So lastly, let's just address what's coming up. We have an upcoming data release later on this year, which is super exciting. We also have this study that is currently ongoing with the CAR-T. And then there's an analogous CAR-T that's being generated by NCI. This is for autologous cells, and that data is also expected soon. Lastly, big question. How do we survive this downturn? Well, I can't answer this because I would be a billionaire if I did, and I wouldn't be talking here. But what I can tell you is that what we did at Vor is we firstly raised as much as we could when we could. Now, that's easy to say, harder to do, but something that we just kind of scrub every opportunity that we can to raise money. And that needs to be done on the basis of, I'm sorry, if you can advance the slides. On the basis of clinical catalysts. Now, what if you don't have clinical catalysts? Then I think it's really focusing on the science that could generate a clinical program, and that might involve certain difficult prioritization decisions. But that's the environment we live in today. And really, the market doesn't really like companies like mine, which are early clinical, but it hates even more the preclinical companies, right? So really focusing on generating those clinical milestones and value in the clinic is key. Investor relations is everything. You have to show to investors that you have a plan. It's a robust plan, something that you can execute on, and that you're going to meet the goals and milestones that you promise. That's been super important to me personally, leading Vor, and I think it's shown in terms of the faithful investors in our investor base. But it's something that I think is all the more important in our industry, where all kinds of things can happen. But above all, really try to meet those milestones and focus on value creation. Lastly, I'll just mention and remind you, ARM is meant to be an alliance. It's meant to be an alliance of companies where we're working together, and we know, goodness knows how difficult this is separately, but we need to be working together. Now, we all have different agendas, we all have different things that we are focusing on, but at the same time, we should not hesitate to reach out and help our fellow member. So that includes just advice, it includes just general guidance on matters that may not be pertinent until 6 months from now. We have to focus on just this synergy of us together here, and I think we can help each other as well as help patients in need. I want to thank you for your time.